So since we are done with the genetic polymorphism in drug metabolism, today we'll be talking about drug target and drug transport. So as we saw in the previous class, if there is a polymorphism in any of the gene that encodes for a protein or an enzyme responsible for drug metabolism, then the clinical effect can be different because of that polymorphism. Similarly, here, if any of the polymorphism is happening in drug target, so or drug transport, then the outcome, clinical outcome, will be different. So before I dive into the chapter, I would like to interact with you people about what do you know about drug target. When I say drug target, what does it mean to you?
The receptor which
receptor activity, sir, which
okay, good. This can be one receptor. Okay, what else? Where the drug acts? Target.
any enzyme.
We're talking about drug target. So okay, one target is receptor. Okay, next target could be enzyme. Okay, you know that drug do act on enzyme as well. Okay, so what else you know as a drug target? Channels, very, very important. You know there are many sodium channel blocker, potassium channel blocker. So so that is another target for the drug to act. Okay, so these are the common drug target, and then you have something called drug transport. Okay, so drug transport. When I talk about drug transport, what comes in your mind? What comes in your mind when I talk about
distribution of the drug and
transport
from extracellular to the
look at the word transport. Okay, so we are talking about here
from extracellular molecule
absorption
path.
Yeah, you are right. Okay, extracellular absorption, all those things. But here, there will be the polymorphism in like a transport level. When I'm talking about transport level, that means the polymorphism. There is because of the polymorphism, there is the effect on transport of a drug, isn't it? So what can be the effect of what effect can be there of polymorphism by affecting the transport? That means either the drug is increasing or decreasing, isn't it? Doesn't you get a sense of that? So that might be happening again because of that. You can figure out that might be happening again because of the transporter involved in it. So here, when I say drug transport, we are talking in more detail about we are referring to drug transporter. Again, you know there are many kind of drug transporter, right? Like again, it could be a channel, it could be a protein. Okay, so that means today we're going to talk about genetic polymorphism in this drug target. Whatever you told, listed out like ion channels, enzymes, receptors, and genetic polymorphism in drug transporter. Okay, because of that, the drug concentration may get changed. Clear with the introduction, everyone? Okay, so the first have you heard about this P-glycoprotein?
So in drug interactions.
What is this? Is this I think you have heard many times because you might have come across a drug interaction which have affected this P-glycoprotein, and then either there is increase or decrease. So many times you might have come across this, but have you ever given a thought? Is it a target or a transporter?
Target, sir.
No, that's not a target. It's a transporter. Okay, Srikant? This is a transporter. I'll explain you how. Okay, so P-glycoprotein and MDR. So what does MDR stands for?
Multi-drug.
Yes.
Multi-drug receptors.
Multi-drug resistance protein. Okay, this MDR multi-drug resistant protein. I will explain why we call it as multi-drug resistant. In the coming section, I will talk about that. But let's start with the intro of P-glycoprotein and MDR. You know, drug gets transported by many transporters, as I talked. One of the transporters is this P-glycoprotein. This P-glycoprotein is present abundantly in the gut lumen, in the brain, in the brain, like in the like nearby blood-brain barrier, like testis blood barrier, placental barrier, in the biliary and urinary like excretion. These help, pig glycoprotein helps. So these are the like common places where this pig glycoprotein are present in abundant quantity. So that means many of the interaction that you have seen was happening at this one of these sites. So again, every individual is different, and because of that, all this polymorphism and all this amount of the drug target or the transporter differs. So this is the general introduction of about pig glycoprotein, where it's present. But what it does actually is, let's also see what it does. It is a efflux pump. In simple layman language, you can understand it is a pump which push out the things from inside to out. Okay, that's it is called efflux pump. And to push out the things from in to out, this needs ATP. Okay, that's why you call it as ATP dependent efflux pump. Okay, so why do we need that? Actually, we need this is pig glycoprotein is very much essential and very much required for our body, because this helps in efflux of the toxin, which is not required in the body. So we don't need those toxins in the body. So these toxins gets excreted or gets pumped out of the cell through this pig glycoprotein. Okay, now we'll see the implication of this in the drug. Okay, because of course drugs are also not a normal endogenous physiological substances. These are also xenobiotics. Okay, these are also kind of toxin to the body. So the pig glycoprotein will also efflux those drugs out of the cells. So that is the impact or implication that we are going to focus in this topic. So now, before I go further, let us have the grasp of this also. Okay, now you understood that there is the pig glycoprotein whose function is to pump out the drug. So how clinically this gonna be impacted? Whether the effect gonna increase with more pig glycoprotein, effect of drug, or the effect of drug is gonna be decreasing with more pig glycoprotein. What do you say, Rowena?
Yes, sir.
Did you listen my question?
No, sir.
Vinushree? Vinushree?
Like how it can affect.
Pratyush? Pratyush?
Yes, sir.
Did you listen
Yes,
my
sir.
question?
Yes, sir.
What was that? Repeat that. Let everyone know.
Like protein will affect the transport. It will increase or decrease the effect.
Okay, you repeat once more, because many people didn't hear because of unstable internet. You repeat that again.
Will P glycoprotein will increase or decrease the effect of drugs?
Yeah, did you get the question, Rowena, now? See,
Yes.
please pay attention here, because you are not physically present here. That doesn't mean you log in and go somewhere else. I'm telling this to everyone. Okay, I can ask randomly question to you. If you are not responding to that, I might be absent. That's all we can do. That means that you are not listening to us. Okay, if I ask some random question and you are not able to reply, so please pay attention. So what I asked is, please mute yourself. It's noisy little bit. So what I was asking is, now you understood the function of pig glycoprotein. So I'm asking, if pig glycoprotein is more, the effect of drug going to be increased or decreased? Very simple, very easy question.
Sir, I think it depends whether it's in the that pig glycoprotein is in the absorption point of the drug, or else like the.
I told you where they are. I told you their location very clearly, right?
So then it like if
bioavailability is ready.
Yeah, it's very simple. There is nothing to get confused. Very easy. I told you this the primary function of this P glycoprotein was to pump out the toxin, to efflux the toxins. But they consider even drugs as xenobiotics, because they are the xenobiotics and they are considered as toxins for the body. This P glycoprotein even efflux or throw out the drugs. So it's very easy. That means if the P-glycoprotein is more in the that border area, whether it is brain or testes or fetus or the intestine, that means the drug is getting out of the cells in a more quantity. That means the biology is decreasing, as Sarath said rightly. The drug concentration in the blood gets decreased. That means the drug is not going to act more effectively, right? There can be therapeutic failure. Clear? Very easy, right? Okay, so now you got to know the implication of either more P-glycoprotein or less P-glycoprotein.
Your voice is keeping on breaking, sir.
Is it?
Naresh, sir,
Sir,
your
your
voice
voice
is
is
keeping
not
on
clear.
breaking.
Okay, then let me see if I can switch to the network. Okay, so I was talking, if the P-glycoprotein increases, what happens to the biology? If the P-glycoprotein decreases, what happens to the biology? So with this knowledge, now we'll move forward in knowing there are P-glycoprotein inhibitors and there are P-glycoprotein inducer. So there are some substances that can increase the P-glycoprotein, thereby decrease the biology and decrease the efficacy, that is therapeutic failure. And there are some substances which inhibit the P-glycoprotein, thereby decreasing the efflux, thereby increasing the biology, and thereby increasing the toxicity or therapeutic effect. Okay, so this is the concept that you should know, and even in your drug interaction. So do you remember? Are you noting it that many things from drug interaction chapter is related to pharmacogenetics? We saw even in the previous drug metabolism cases, drug metabolism chapter, where we are talking about cytochrome 450 enzyme and many other enzyme where the drug interacts by depending on such enzyme. So all these enzymes, all these transporters are related to gene actually. Okay, that is why the genetic information is very much important when you extrapolate. When you extrapolate or when you try to extrapolate the theoretical interaction to clinical significance. Okay, so you cannot do that alone. So that is why I keep telling you people in the ward or in the hospital that finding theoretical interaction means nothing, absolutely nothing, unless you don't have the peripheral information about patients, about other drugs, about many other conditions. So you should have many information to infer something about interaction, therapeutic drug interaction. Okay, so recently there are several polymorphisms that have been identified in this gene. So MGR1 is a gene. Okay, multi-drug resistant one is a gene. I will tell you why this is called multi-drug resistant. And the same gene encodes for a protein. That protein name is Priya. The protein that is encoded by the gene MGR1, the name of that protein is? Anybody? Anybody?
MDR protein.
That is P-glycoprotein. Okay.
P-glycoprotein.
P-glycoprotein.
Okay.
Protein.
Yeah. So I will tell you. Okay, the other name of MGR1 gene also I'm going to tell you in the coming slides. So you should know the function of this gene, MGR1 gene encodes for P-glycoprotein, and the function of this P-glycoprotein and the relation of this function in terms of the interaction. So as now, if there is a polymorphism in MGR.
Sir, I can't hear you.
We didn't hear anything.
Okay, and now is it better now? I stopped my video. Is it any better now?
Hello.
Sir, we can't hear you.
So now we will be looking what happens to the final effect that we saw by increase or decrease in P-glycoprotein function when there is some sort of polymorphism in this gene, MGR1 gene. Okay, so we all know that now this is responsible for efflux of the drug. That is, in other words, kinetic word, we can tell it as drug disposition. Okay, drug disposition, and therefore there are some risks with this that we know something and we will discuss here. So including all these things, when I say all these, that means MGR1, P-glycoprotein. So this is called transporter pharmacogenetics, because we are dealing right now with the transporter. Okay, these are the substances, these are the proteins that helps in transport of a drug, and we are also discussing about the gene that encodes for such protein. So these are collectively called as transporter pharmacogenetics. Okay, so this transporter pharmacogenetics is concerned with the uptake, okay, and the efflux of the drug to and fro. And based on this, you can have many clinical inference, okay, because this is directly related to biology of the drug. And you also know that I have told already in the beginning of this chapter that there are several membrane transporter proteins that are involved in the absorption of drug from the intestine into the body, and also from the non-intestinal places. So now you will understand why I was talking. The name of that protein is, or name of that gene is MGR1 gene, multi-drug resistant. Since the drug is going out, so that means the drug is not showing its adequate effect. Am I right? So that means the effect that we expect from the drug is not being shown because the therapeutic concentration is not maintained because of efflux. So because of that, the resistance is happening. If you remember from even from your microbiology or pharmacology, there were several mechanisms of resistance, and one of them was this efflux pump present even in the bacteria. That doesn't allow the drug to go into the bacteria, and we cannot kill the bacteria. So same thing applies even here. So here the drug is not getting into the required tissue wherever we want, and we are not getting the effect. So that in other words, this is again a resistance. Okay, the drug is not acting on the patient. So this was actually seen previously with some of the anti-cancer drugs like vinblastine, vincristine, doxorubicin. So those drugs, the patient was not responding, and later when they studied the reason, they found out this is because of this MDR1 gene polymorphism. Okay, so there are many multi-resistance protein, and these are the member of ABC superfamily. ABC means ATP binding cascade. Okay, so ABC means ATP binding cascade, and again in ABC you have many ABC1, ABC. This is superfamily. Again, you have many family in it. Okay, so if you remember, one of the genes that we are studying for levetiracetam, that we are studying still for levetiracetam, is ABCB1 gene. Okay, so there ABC is nothing but ATP binding cascade, and B1 is simply the name of that protein, okay, or that gene in the sequence. So there are many MRP, multi-resistant protein. Again, MRP is one of the member of the superfamily ABC. Okay, and there are 6 members of this multi-resistant protein. So out of these 6, the 3 are very commonly known to affect the drug disposition: MRP1, MRP2, and MRP3. So drug transporter are one of the important reasons for this. Yes, Gayatri? Gayatri raised your hand.
Can you go? Yeah, can you go a little slow, sir?
Okay. Okay, so drug transporter are now increasing. This transporter pharmacogenetics is a growing field now, because we now are figuring out that there are many drugs that depends on this transporter, and because of that, either the patient is not getting the response, and or the patient is getting enough like exaggerated pharmacological action, that is side effect. That is what you read in many kind of drug interaction. Okay, so I told you why this is called multi-drug resistance, because it was involved in resistance of many anti-cancer drugs, and I was talking because of this efflux transporter, how it going to impact the drug interaction. Okay? Sometimes it can cause therapeutic failure, sometimes it can cause toxic effect. So I was telling, I will tell you another name of this MDR1. So the another name is same, and ABCB1 gene. Okay, so either you say MDR1 gene or ABCB1 gene. That means same thing. You can get confused with that. So recently, a number of SNPs in MDR1 has been identified. That's why this is a growing field now. And I was telling, actually, if you want, you can design a study, research study in your hospital by, like, of course, it needs fund, but that's going to be a good study. Like you are dealing with the interaction, and you want to see the effect of interaction involving P glycoprotein, and you try to do the TDM to set the or to estimate the drug concentration in the blood by doing a TDM, and then because you know that if you want to know the significance, how clinically significant the drug interaction is by doing a kinetic. Okay, so this is going to be interesting study if you design such study. Like you have the theoretical evidence that this in this interaction P glycoprotein is involved, and P glycoprotein is involved, and the drug is getting out of the cells. If out of the cell, where it will go? It will go in the blood, right? So you are doing the TDM to check the drug concentration in the blood. So if you design any such study involving any drug that is very much related to this P glycoprotein dependent interaction, would be a nice study. That's what I wanted to tell. So an increase in number of study, that's why there are many studies now increasing. And this have impact in Parkinson's disease, inflammatory bowel disease, refractory seizure as well. Okay, that is why we without to choose this levetiracetam drug and then see what is the efficacy of this drug, because in case of refractory seizure also this P glycoprotein have major role. You know, refractory seizure is nothing but what is that? Pashupati, can you repeat it to everyone?
Status epilepticus.
No. Pashupati, what is refractory seizure? Can you guys listen me smoothly?
Yes, sir.
Yes, Pashupati, I asked the definition of refractory seizure.
Yes, sir. When in refractory seizure, then there use of 2 or more than 2 drugs.
Okay,
antiepileptic drugs.
So what you want to tell is that with the two, like with the one drug itself, or with the two drug, the epilepsy is or the seizure is not being controlled. So you have to use at least 2 or more than 2 drugs in such seizure. That's what refractory seizure, isn't it, Pashupati?
Yes, sir.
So what does this mean again? This means that the drug is not, the initial drug that you are giving is getting efflux and it's not working. Okay, so that is why you the refractory seizure is happening. The seizure is going to refractory not responding to the treatment. So refractory seizure, Parkinson's disease, inflammatory bowel disease, even the CD4 cell, CD4 cell in a HIV patient during recovery. So all these phenomena, all these condition uses this P glycoprotein. So, however, in many such cases, the reported effects of MDR1 or P glycoprotein SNPs.
Can't hear you, sir.
Sir, can't hear. Hello.
Yes. Can you hear me? Can you hear me? Can you hear me, guys? Can you hear me?
Sir, we can't hear you, sir.
Okay, let's schedule at 4 to 5.
Yes, sir.
Okay, so I told you why this is called multi-drug resistance, because it was involved in resistance of many anti-cancer drugs, and I was talking because of this efflux transporter, how it's going to impact the drug interaction. Okay? Sometimes it can cause therapeutic failure, sometimes it can cause toxic effect. So I will tell you another name of this MDR1. So the another name is same, and ABCB1 gene. Okay, so either you say MDR1 gene or ABCB1 gene. That means same thing. You can get confused with that. So recently, a number of SNPs in MDR1 has been identified. That's why this is a growing field now. And I was telling, actually, if you want, you can design a study, research study in your hospital by, like, of course, it needs fund, but that's going to be a good study. Like you are dealing with the interaction, and you want to see the effect of interaction involving P glycoprotein, and you try to do the TDM to set the or to estimate the drug concentration in the blood by doing a TDM, and then because you know that if if you want to know the significance, how clinically significant the drug interaction is by doing a kinetic. Okay, so this is going to be interesting study if you design such study. Like you have the theoretical evidence that this in this interaction P glycoprotein is involved, and P glycoprotein is involved, and the drug is getting out of the cells. If out of the cell, where it will go? It will go in the blood, right? So you are doing the TDM to check the drug concentration in the blood. So if you design any such study involving any drug that is very much related to this P glycoprotein dependent interaction, would be a nice study. That's what I wanted to tell. So an increase in number of study, that's why there are many studies now increasing. And this have impact in Parkinson's disease, inflammatory bowel disease, refractory seizure as well. Okay, that is why we without to choose this levetiracetam drug and then see what is the efficacy of this drug, because in case of refractory seizure also this P glycoprotein have major role. You know, refractory seizure is nothing but what is that? Pashupati, can you repeat it to everyone?
Status epilepticus.
No. Pashupati, what is refractory seizure? Can you guys listen me smoothly?
Yes, sir.
Yes, Pashupati, I asked the definition of refractory seizure.
Yes, sir. When in refractory seizure, then there use of 2 or more than 2 drugs.
Okay,
Antiepileptic drugs.
So what you want to tell is that with the two, like with the one drug itself, or with the two drug, the epilepsy is or the seizure is not being controlled. So you have to use at least 2 or more than 2 drugs in such seizure. That's what refractory seizure, isn't it, Pashupati?
Yes, sir.
So what does this mean again? This means that the drug is not, the initial drug that you are giving is getting efflux and it's not working. Okay, so that is why the refractory seizure is happening. The seizure is going to refractory not responding to the treatment. So refractory seizure, Parkinson's disease, inflammatory bowel disease, even the CD4 cell, CD4 cell in a HIV patient during recovery. So all these phenomena, all these condition uses this P glycoprotein. So, however, in many such cases, the reported effects of MDR1 or P glycoprotein SNPs.
Can't hear you, sir.
Sir, can't hear. Hello.
Yes. Can you hear me? Can you hear me? Can you hear me, guys? Can you hear me?
Sir, we can't hear you, sir.
Okay, let's schedule at 4 to 5.
Yes, sir.